Now we'll go on to Russ Kibbe from North Carolina State University, who has been working in the lab of Dave Modiman, who you may know as the father of MSiReader. And Russ will talk about MSiReader and the latest development and how it can be used with AP MALDI data.
All right, hi, good morning everyone. Like he said, I'm from NC State University. I work under David Muddiman, and this morning I'm excited to talk to y'all a little bit about MSiReader, some of the things that it can do, and then show some examples on some AP MALDI data using the MassTech AP MALDI source. So here I've listed just a few of the things that MSiReader can do. These are just a few of the capabilities. I'm going to go through some of these in some more detail, and like I said, show some more examples. So first, an introduction to quality control system suitability testing. This is a project that I've been working on, really working with MSiReader and helping to improve it. There's currently, quality control is the operational processes and techniques that we use to evaluate results, and system suitability testing is how we evaluate the analytical performance of our instrument. This is really important to understand the state of the system, ensure repeatability, reproducibility, and these protocols are essential to important experiments and investigations. One area where this is lacking is in mass spectrometry imaging, where there's currently no standardized practice or protocol, even though this is a very important issue. And so we're setting out to try to create a commercially available mixture and a process to be able to quickly assess the state of the system. To do this, the approach we've taken is a machine learning approach, where we're using our novel QC mix, collecting that QC data. We then can use the capabilities of MSiReader to automatically extract these QC relevant metrics such as monoisotopic and isotopic abundance, MMA, relative standard deviation, detection frequency within the ROI, and the spectral accuracy of the peaks within that QC mix. We then format the data and apply those class labels, whether the instrument is clean or compromised, split the data into training and testing sets. For this validation study, for actual implementation, you would use this entire set as the training set, and then insert your single ROI for testing. We then scale the data based on the training set and then provide the data to multiple different supervised machine learning models and obtain the instrument classifications from that. And so this is all using the Auto MSI QC tool that is present in MSiReader. Some of the other statistical analysis that's available in MSiReader, I have here loaded two ovarian tissues. One is cancerous on the left and healthy on the right. We can draw ROIs around both of those tissues individually, one being the interrogated ROI and one being the reference ROI. But then selecting both of those ROIs, we can automatically export the abundance data of these two different tissue types into an Excel or a CSV file like this, where you're getting pixel by pixel or voxel by voxel abundances at each one of the M/Z values within a given list, whether that list comes from an internal list or a database that you're using for search. We then can utilize this data export to do some powerful statistical analysis such as PCA or principal components analysis. And so using the built-in function of MSiReader, you upload that data export, provide some parameters, and you'll get this principal component analysis output where hopefully you're seeing grouping of your cancerous scans and your healthy scans, or at least scans between your two experimental groups. Not only do you get this nice PCA output, but you can also dive deeper into it, looking at the different loadings for the PCA analysis, as well as interact and view the different scree plots that are produced for all the different features within your data set. And so we have highlighted here the glutathione signal and its loading for this specific PCA analysis. We can also do T-distributed stochastic neighbor embedding, or TSNE. A similar process, you export the data between your different groups, provide the necessary metrics, and then again you get this output grouping the different experimental groups. This is just for a single cancerous tissue and a single healthy tissue, so you can imagine as you go get more biological replicates that this could get very noisy. And so one of the capabilities within TSNE is that we can actually reduce those clouds down to an average point, so as you increase the number of biological replicates, you're seeing you'd have a more informative cloud of average values rather than just a mess of scan values when you have tissues that are on the orders of thousands of scans. And so now to dive into some of the AP MALDI data that we've been working with, we're looking at, or we've seen vasopressin in the pituitary adenoma here at a 5 micron spatial resolution. And you can see the really fine detail that you get in that image. And then on the other image, looking at dopamine in the hippocampus at a 25 micron spatial resolution. We're able to visualize these really well and using the Civitas color map to really show the abundance values and the chemical localizations within the tissue. Not only is the detail of the images really great, but the quality of the data is also really great. Looking at the MMA heat maps that you can pull out of MSiReader, the yellow color shows at 0 ppm mass measurement accuracy, and so by the overwhelming yellow color shown on tissue, we're seeing sub ppm mass measurement accuracy for both this vasopressin and dopamine when on tissue. This is using easy IC on and with the AGC disabled, using the AP MALDI source coupled to the Explorers 480. Another advantage that we can use with the MSiReader platform is this co-localization algorithm, or the SSIM tool as we call it, where you provide a reference image that can also be an optical image or an ion image as shown here, and then looking through a list of compounds or peaks, you can automatically find similar images or even perhaps detect images that are much different than your reference image. So an example of how we would do this, looking back at that hippocampus tissue, looking at the reference image, we have the guanine shown in the upper left. And so you would upload a list of peaks to the MSI correlation parameter selection screen, such as, again, an internal list or a list that you obtained from using database annotation. You would upload that list, or provide it the reference MRZ, like I said, we're using guanine here, and then it would give you SSIM scores output as well as heat maps that coincide with those different peaks that you provided. And so you can see in the bottom left, there are 2 peaks at the top that have a really high similarity, and those are also shown as heat maps in the top right. So you can see that the chemical localization for these different peaks localized in the same area as guanine, which can be informative to future investigations. But then at the bottom, we also have an SSIM score that's pretty low, showing a different localization for another compound, which again can be very informative and helpful going forward with future investigations. Also, a new and exciting part of MSiReader is the biopharma option. This is specifically suited to high throughput screening and phenotype analysis and phenotype screening using well plates, looking for hits. And so MSiReader the biopharma mode, comes with lots of options for analyzing well plate analysis. And what you're seeing here is the heat map where each square, each voxel shows data from an individual well plate or a well in the well plate. Where each spot is an average of 3 shots. And so this is a really quick way that we can analyze well plate analysis and analyze hits for pharmaceutical applications. And with that, I'd like to thank everybody at MassTech, Proteoformics, and LIST, and for the opportunity to speak to you all this morning. Thank you.